If you've ever opened a research paper and thought, I want to understand this, but this is dense, then this podcast is for you. Every day, thousands of research papers are published. Most of them are rigorous, insightful, and almost impossible for non-specialists to read comfortably. But the ideas inside them shape medicine, technology, business, and society. Welcome to One Paper Orbit. We are a research paper podcast. Here we take peer-reviewed research and turn it into clear, structured conversations without adding speculation, without exaggeration, simply making the original work easier to understand. Today we are discussing a paper authored by Zachary S. Lorsch and Roger A. Little titled Mechanisms and Clinical Implications of Gut-Brain Interactions, published on January Tech 2026 in the Journal of Clinical Investigation. This discussion is based on a paper published under the Creative Commons Attribution 4.0 license. The original authors retain credit for their work. This podcast provides an adapted explanation for educational purposes with full citation and license information provided in the description. So we really have an incredible mission for this deep dive today. We're going to explore the biological superhighway that connects our stomachs directly to our minds. I mean, have you ever had a gut feeling about a decision? Or felt those actual physical butterflies in your stomach right before a big meeting?
Right, or before a first date.
Exactly. We are going to examine the literal, hardwired anatomy that makes those sensations happen, because our gut and our brain are in this constant high-speed conversation that dictates, well, pretty much so much of our daily lives, from our mood to our immune system and even our choices about what we eat.
It really is a profound shift in how we view human biology. For a long time, modern medicine heavily compartmentalized the body. You went to a neurologist for your brain, a gastroenterologist for your stomach, a psychiatrist for your mood.
Like separate mechanics for different parts of a car?
Exactly like that.
Huh.
But what the research over the last decade has demonstrated is that these systems are physically and chemically inseparable. It's fascinating to look back and realize that over 2,000 years ago, the ancient Greek physician Hippocrates famously suggested that all diseases begin in the gut.
Which sounds like a broad philosophical statement at the time.
Right, but science has finally mapped the exact physical mechanisms, proving that he was remarkably close to the truth. The interactions between the digestive system, the microbiome, and the brain are now implicated in a vast array of human health conditions.
Okay, let's unpack this history, because the way we started figuring this out is full of these interesting little breadcrumbs that hinted at this connection way before we had the technology to actually map it. There's this famous case from 1833 involving a physician named William Beaumont.
Ah, yes, the Beaumont case. It's a classic.
Right. He had a patient who had suffered a severe abdominal wound, and it healed with a permanent opening, a fistula, directly into his stomach. So Beaumont could literally look inside the man's stomach. And he made this profound observation. He discovered that when his patient experienced emotional arousal like anger or frustration, it visibly and directly altered his gastric secretions.
In real time, which was a revolutionary concept. Beaumont essentially had a window into the physiological effects of emotion. And then decades later, you have Ivan Pavlov.
The bell and the dogs.
Exactly. Most of us are familiar with his work on classical conditioning, ring the bell to elicit salivation. But his Nobel Prize-winning work actually detailed the cephalic phase of digestion.
Cephalic meaning head. Right.
Yes, the physiological process where just the thought, sight, or smell of food prompts the brain to send signals down to the stomach. It stimulates the release of gastric juices long before a single bite of food is ever consumed.
So the brain is essentially prepping the stomach for an event that hasn't even happened yet.
That's it exactly.
Right.
Beaumont and Pavlov proved that a physiological connection existed. But knowing a connection exists and knowing precisely how that connection operates on a cellular and molecular level are two very different things.
Which is the core of our deep dive today.
It is. We are looking at the exact defined signaling pathways. And to really grasp how these signals are sent and received, we first have to understand the nervous system of the gut itself. It is so remarkably complex that neurologists and gastroenterologists frequently refer to it as the second brain.
I think most of us, well, I know I used to, just picture our intestines as a biological tube. Just a passive plumbing system squeezing food along.
A lot of people do.
But that couldn't be further from the truth. We're talking about the enteric nervous system, or the ENS. The sheer diversity of cells lining this system is staggering. The paper details that there are 21 distinct neuronal subtypes and 3 distinct glial cell subtypes right there in the tissue of your gut.
To put that into perspective, your spinal cord doesn't even have that level of intrinsic localized complexity in terms of varied standalone functions. The basic architecture of this enteric nervous system is divided into two primary layers, or plexuses.
Embedded within the walls of the digestive
Yeah.
tract, right?
First, you have the submucosal plexus. You can think of this layer as a chemical and sensory manager. It is primarily in charge of controlling mucosal processes, so it regulates the secretion of digestive enzymes, the absorption of nutrients across the intestinal wall, and the local blood flow required to fuel those processes.
And the second layer handles the physical movement.
Precisely. The second layer is the myenteric plexus. It controls the actual mechanical movement, the coordinated muscular contractions known as peristalsis that propel contents through the digestive tract.
And supporting both of these neuronal layers are the glial cells.
Right. And historically, we thought glial cells were just the structural glue holding neurons together. But we now know they are highly active participants. They refine and coordinate the activity of the neurons, manage local inflammation, and help maintain the integrity of the gut lining.
There is an incredible demonstration of just how autonomous this second brain really is. If you were to take a section of the colon and physically remove it from the body,
Ex vivo.
right, completely severed from the brain, the spinal cord, everything, it can literally still perform peristalsis. It can continue that rhythmic squeezing motion in a laboratory setting. It doesn't need the central brain to tell it how to do its primary job.
It has its own localized, independent intelligence. It is capable of entirely autonomous function for those basic rhythmic movements. However, in a living, breathing human body, it is never operating in isolation.
It's always talking to the rest of the body.
Exactly. The enteric nervous system is intimately wired into the autonomic nervous system. And crucially, if we connect this to the bigger picture, this communication is completely bidirectional. It's not just the brain barking orders at the stomach.
So the stomach is talking back.
Loudly. For instance, there are specialized neurons in the gut called viscerofugal neurons. Unlike typical motor neurons that receive commands, these viscerofugal neurons project their axons out of the gut. They connect directly to the sympathetic ganglia.
Those are the nerve clusters outside the spinal cord that control our fight or flight responses, right?
Yes, so the gut is actively sending status reports out to the rest of the nervous system. It creates a continuous dynamic feedback loop. The gut tells the sympathetic nervous system what it's currently experiencing.
Like maybe it's distended or it detects an irritant.
Right, and the sympathetic nervous system processes that data and sends signals right back to modulate the gut's motor activity based on that specific information.
Which naturally brings us to the absolute star of the show when it comes to the gut-brain axis, the vagus nerve. If you imagine all these local gut neurons as a bustling city network, the vagus nerve is the massive, high-speed fiber optic cable linking that city directly to the central command center in the brain.
The primary data superhighway.
Exactly. And the name vagus actually comes from the Latin word for wandering, because it exits the brainstem and wanders all the way down through the chest into the abdomen and branches out to innervate the entire length of the gastrointestinal tract.
Heavily concentrating on the stomach and the upper intestines. The vagus nerve, working in tandem with the pelvic nerves and various sensory nerve fibers that run through the spinal cord, forms an incredibly dense web of communication.
Reaching right down to the inner lining of the gut.
The epithelium, yes. They act as sophisticated sensory antennae. They receive chemical signals and neurotransmitters from the gut, encode that information into electrical impulses, and fire those impulses straight up into the central nervous system.
So the brain is constantly, every millisecond of the day, receiving highly detailed status updates about the chemical and physical state of your digestion.
Every single millisecond.
So if this network is firing lightning-fast electrical signals back and forth all day, how is the actual data packaged and sent? Because it's not just one type of signal. The communication between the gut and the brain essentially relies on 4 major pathways or pillars of communication.
And the first pillar completely changes how we think about hormones.
Right, because most people, when they hear the word hormone, they immediately think of the thyroid gland or the adrenal gland sitting on top of the kidneys or the reproductive system. But the gut is actually the largest endocrine organ in the entire human body. It secretes more than 30 individual hormones.
It operates as a massive, continuous hormonal factory. This signaling originates in specialized cells scattered throughout the lining of the gut, called enteroendocrine cells, or EECs.
And structurally, they don't even make up that much of the gut lining, do they?
Only about 1% of the cells lining the gut, but their physiological impact is immense. They are categorized and defined by the specific hormones they manufacture, and this distribution varies widely depending on whether they are located in the stomach, the small intestine, or the colon.
So they're like microscopic taste buds lining your intestines.
That is a perfect way to visualize it. They literally taste or detect the chemical composition of the food you just ate, the fats, the sugars, the proteins. And in response, they release specific hormones either systemically into your bloodstream or locally into the surrounding tissue to coordinate the digestive process.
Let me make sure I'm getting this. When I eat a meal, these enteroendocrine cells are chemically analyzing it on the fly and pumping out hormones to tell the rest of my body what to do with it.
Precisely. They orchestrate everything from telling the pancreas to release insulin, to signaling the gallbladder to release bile, to ultimately telling your brain when you have had enough to eat.
And this is where we run into one of the most fascinating paradoxes in human biology, specifically regarding serotonin. We all know serotonin as a crucial neurotransmitter in the brain that regulates mood, sleep, and anxiety.
The target of many antidepressants.
Right. Millions of people take SSRI antidepressants specifically to keep more serotonin active in their brain circuitry. But here is the catch. 90% of the body's total serotonin is manufactured down in the gut. So the logical leap is to think, great, if I eat foods that help my gut make more serotonin, that serotonin will travel to my brain and make me happier.
So it doesn't.
Exactly. Gut-derived serotonin does not cross the blood-brain barrier. It is physically blocked from entering the central nervous system. So how on earth is this massive pool of gut serotonin affecting our minds if it can't even get in?
This is a major point of misunderstanding in popular science, and it requires us to look closely at how peripheral hormones bypass or interact with the brain security system. The blood-brain barrier is a highly selective, tightly packed layer of cells designed to protect the brain from toxins and pathogens in the blood.
A microscopic bouncer.
Exactly. While it is true that serotonin itself is too large and lacks the specific transport mechanisms to cross, other gut hormones, like ghrelin, the hunger hormone, and leptin, have dedicated transporter proteins. They are essentially granted VIP access to cross the barrier.
But for the hormones that cannot cross, like serotonin, the body has evolved brilliant workarounds. How does a hormone deliver a message to a building it isn't allowed to enter?
One primary method is through structures known as circumventricular organs. A key example is the area postscrema, located at the base of the brain. You can conceptualize these areas as specialized security checkpoints where the blood-brain barrier is intentionally designed to be somewhat permeable or leaky.
So it lets things through locally.
It allows peripheral hormones circulating in the blood to bind to receptors on brain cells located in these specific zones without having to cross the tight barrier into the deeper brain tissue.
It's like a highly secure government facility that has a specific reinforced window near the entrance. You can walk up, hand your message through the slot to the guard, and the guard takes inside, but you never actually step foot in the building yourself.
That is an excellent analogy. The area poststrema is actually known as the brain's vomiting center. It samples the blood for toxins and triggers nausea if it detects them, which is a direct gut-brain protective mechanism.
That makes total sense.
Furthermore, hormones circulating in the bloodstream can interact directly with the cells that make up the blood-brain barrier. They bind to the exterior of these barrier cells, causing the barrier cells themselves to release secondary messenger molecules, like inflammatory cytokines or nitric oxide, directly into the brain tissue.
So the hormone knocks on the outside of the door, and the door itself shouts the message to the inside. And there's a third workaround, right? The hormones don't even need to travel up to the brain via the blood if they just talk to the vagus nerve directly.
Exactly. The gut serotonin and other hormones bind to specific receptors located on the sensory endings of the vagus nerve, right there in the gut lining. The vagus nerve then instantly translates that chemical hormonal signal into an electrical impulse that races up to the brainstem.
Which perfectly transitions us into the second major pathway of communication. Here's where it gets really interesting. Because for a very long time, scientists thought this hormonal drift, releasing chemicals into the blood and waiting for them to circulate, was the primary way the gut communicated.
What researchers recently discovered a completely new type of cell that revolutionized our understanding of this system, the neuropod cell.
Neuropod cells.
The discovery of neuropod cells is one of the most significant breakthroughs in neurogastroenterology in recent years. We mentioned the enteroendocrine cells, the ones that release hormones. Well, researchers discovered that a specialized subset of these cells possess a remarkably different physical structure.
They aren't just round cells.
No, instead of just being round cells that dump hormones into the local environment, these cells have long physical extensions, almost like the arms of an octopus called podocytes. And these extensions reach deep into the gut tissue to form physical synaptic-like connections directly with the nerve terminals of the vagus nerve.
So they are physically hardwired together.
Yes, they possess all the necessary biological machinery for true, rapid neurotransmission. This fundamentally changes the timeline of gut-brain communication.
Because hormones are slow.
Hormonal signaling, by its very nature, is a slow process. A hormone must be synthesized, secreted into the interstitial fluid, absorbed into the capillaries, circulated through the entire cardiovascular system, and eventually bind to a specific receptor. That can take minutes to hours.
But neuropod signaling is electrical.
It is a direct hardwired connection from the lumen of your gut straight to your brainstem. It operates in milliseconds.
It's the difference between sending a letter through the postal service and sending a text message. It is instantaneous. And what fascinated me is that another large subset of these excitable gut cells, the enterochromaffin cells, actually use serotonin not as a slow-moving hormone drifting in the blood, but as a rapid-fire neurotransmitter across these synaptic connections.
Yes, they use it to instantly modify the electrical signals being sent up to the brain. The sheer speed of this transmission is vital for survival.
It lets the brain react immediately.
Exactly. It allows the brain to make immediate, real-time physiological decisions based on what you are currently ingesting. It conveys highly specific nutritive information, whether a food is rich in fats, sugars, or proteins, from the proximal regions of the gut, like the stomach and the duodenum, straight to the brain's reward and satiety centers while you are still eating.
Okay, so we have the slow systemic hormonal network, and we have the lightning-fast hardwired neuropod connections. The third pillar of communication introduces a fascinating third party into this conversation, the microbiome.
The trillions of bacteria living inside us.
Right, we constantly hear about them, but here they are framed not just as passengers, but as active middlemen, intercepting and modifying the messages between our gut and our brain.
The role of the microbiome in this axis is absolutely critical. We have to view the bacteria in our gut as a massive, metabolically active endocrine organ in their own right. They do not just passively sit there breaking down our food. They're constantly interacting with our biology by producing their own specific chemical byproducts.
And a prime example of this involves short-chain fatty acids, or SCFAs.
Correct. When you eat dietary fiber, complex carbohydrates that human enzymes cannot digest, the bacteria in your colon ferment that fiber. In the process of fermentation, they release SCFAs like butyrate, propionate, and acetate.
And these bacterial byproducts aren't just waste, they are biologically active signaling molecules.
Precisely. These specific short-chain fatty acids can directly bind to receptors on the enteroendocrine cells lining the gut. When they bind, they trigger the gut to release its own hormones, like GLP-1, which regulates blood sugar and appetite.
So the bacteria are essentially pushing the buttons on our gut cells.
They are. They can also interact directly with the immune cells residing in the gut lining, modulating inflammation. And incredibly, some of these microbial metabolites can even cross the gut barrier, enter the bloodstream, and eventually cross the blood-brain barrier to directly influence neurodevelopment and behavior in the central nervous system.
It's like having an incredibly powerful lobbyist living inside your digestive tract, actively pulling levers to change the hormones and signals your body produces based on what you feed it.
That is exactly what is happening. The microbes are entirely capable of inducing hormone release, altering cytokine signaling, and even producing their own precursors to neurotransmitters that modulate the entire communication pathway.
So it's a chain reaction.
Yes, if you change your diet, you change the population of microbes. If you change the microbes, you change the metabolites they produce. And if you change those metabolites, you fundamentally alter the signals being sent to your brain.
Which is a perfect segue into the fourth and final pillar of communication, the immune system. Now, if you spend any time reading health blogs or wellness websites, you have inevitably seen the term leaky gut.
It is a very popular term right now.
It really is, used to explain almost every modern ailment. And while the internet often exaggerates or misapplies the clinical implications, the actual hard biological reality of how the gut's immune system talks to the brain is deeply profound.
The scientific term is intestinal permeability, and it is a very real, measurable phenomenon. Even in a perfectly healthy individual, the gastrointestinal tract is ground zero for immune activity.
Because it's exposed to everything we swallow.
Exactly. It is the largest surface area of the body exposed to the outside world. Because of this, the gut lining is packed with a vast array of immune cells that are constantly surveying the contents of your digestive tract, distinguishing between harmless food proteins, beneficial bacteria, and dangerous pathogens.
It's essentially a massive border patrol operation.
Yeah, that's a good way to put it. Now, when there is a localized inflammatory event in the gut, perhaps due to an infection, chronic stress, or certain dietary irritants, the physical junctions between the cells lining the gut can loosen.
The permeability increases.
Yes. This allows larger molecules and bacterial fragments to slip beneath the mucosal layer, which forcefully activates the resident immune cells. And here's where the gut-brain connection becomes so apparent. This peripheral inflammation in your intestines does not stay local. It is intrinsically mechanically linked to the central nervous system.
There is a specific finding regarding immune cells that completely upends how isolated we think the brain is.
Yes. There is a distinct population of T cells, specialized white blood cells, that are trained and originate in the gut mucosa. During certain physiological conditions or inflammatory states, researchers have documented these specific gut-derived T cells physically migrating away from the gastrointestinal tract.
They actually pack up and leave.
They travel through the systemic circulation, breach the blood-brain barrier, and take up residence deep within the brain, specifically in an area called the paraventricular subfornical organ.
Let's pause and clarify that term, because paraventricular subfornical organ is quite a mouthful. What does that area actually do?
You can think of this specific organ as a central, high-level thermostat and regulatory hub for the brain. It sits near the fluid-filled ventricles of the brain and is heavily involved in monitoring the composition of the blood and regulating the central nervous system's overall balance, or homeostasis.
So you have immune cells that were specifically educated by the environment in your stomach, and intestines physically traveling up to this master control room in the brain to help regulate the brain's internal environment.
Precisely. The gut is sending its own trained regulators straight to headquarters.
That is remarkable. The immune cells are acting as physical couriers, carrying information from the gut's environment directly to the brain's control center. But the communication goes both ways, and there is a study mentioned about a different part of the brain, the insula, that shows just how deep this connection runs.
That particular study is truly a landmark in the field of neuroimmunology. The insular cortex, or the insula, is a region of the brain deeply involved in interoception, which is your brain's perception of the internal state of your body.
It helps you perceive a heartbeat or sense that your stomach is full?
Exactly. Researchers induced a state of colitis, which is severe inflammation of the colon, in mice. Using advanced imaging, they observed that during this gut inflammation, a very specific, distinct population of neurons in the brain's insular cortex lit up and became highly active.
The brain was actively monitoring the inflammation.
Yes. But what they did next is the critical part. They waited for the mice to fully recover. The gut inflammation entirely resolved, and the colon tissue returned to a perfectly healthy baseline.
So the mice were completely fine.
Sometime later, using precise neurological tools, the researchers artificially reactivated those exact same specific neurons in the insula that had lit up during the initial colitis. They just stimulated the brain.
And what happened in the gut?
The activation of those brain neurons sent signals back down the neural pathways that physically recreated the severe inflammation in the colon entirely without any external trigger, infection, or irritant.
Wait, really?
The brain had literally encoded a physical memory of the peripheral immune response, and by recalling that memory, it forced the body to manifest the physical disease state again.
So just by remembering the inflammation, the brain commanded the gut to become inflamed again. It is astonishing. It really drives home the reality that the brain and the gut are essentially a single unified system.
That cannot be separated in clinical practice.
So we have established these four incredible pillars of communication: the systemic hormonal network, the lightning-fast neuropod electrical connections, the microbiome middlemen churning out active metabolites, and the immune system's deep memory pathways.
A perfectly balanced system.
Usually. But what happens when this highly complex system inevitably misfires? Because given how common stomach issues are, this system clearly isn't foolproof. Let's look at how these communication errors translate into real-world digestive diseases.
This is where understanding these mechanisms becomes critical for patient care, because it explains the biological reality behind suffering that was for a long time deeply misunderstood. We see this most clearly in what are now classified as disorders of gut-brain interaction, or DGBIs.
Historically, the medical community referred to these as functional gastrointestinal disorders, right?
Yes, the most widely recognized example being irritable bowel syndrome, or IBS.
The sheer scale of these disorders is massive. Over 40% of people worldwide suffer from some form of a DGBI. If you look around your office or think about your family, nearly half of the people you know are dealing with this to some degree.
It's incredibly prevalent.
And for decades, if a patient with severe IBS pain went to a gastroenterologist, they would undergo a colonoscopy, get an endoscopy, have blood drawn, and all the tests would come back completely normal. The tissue looked healthy. Because doctors couldn't find a visible tumor or an active ulcer, patients were frequently dismissed and told, "Your gut is fine, the pain is just in your head."
Which is a tragic, profound misunderstanding of the biology. In a way, it is in their head, but not in the psychological dismissive sense.
Yeah.
It is a very real, measurable physical malfunction, the communication between the head and the gut.
It's the wiring that's broken, not the pipe.
Exactly. In 2016, the Rome-Tor criteria, which is the international gold standard for diagnosing gastrointestinal disorders, officially renamed these conditions to DGBIs, specifically to recognize this pathophysiology. The tissue might appear normal on a macroscopic level, but the nervous system wiring connecting that tissue to the brain is highly dysfunctional.
Which explains why there is such a massive overlap with mental health. People with IBS or functional dyspepsia frequently suffer from severe anxiety and depression. It's not just that having a stomachache makes you sad. The root pathways are tangled together.
The psychiatric overlap is undeniable, and it heavily involves our fundamental stress response systems. When you experience psychological stress, your brain activates the hypothalamic pituitary adrenal axis, or the HPA axis.
The classic stress response.
Right. The hypothalamus in the brain secretes corticotropin-releasing hormone, or CRH. This hormone cascades down through your system, ultimately prompting your adrenal glands to release cortisol, the primary stress hormone. But CRH doesn't just act systemically, it acts directly on the gut.
Doing what?
Elevated levels of CRH have been shown to immediately increase intestinal permeability, that leaky gut effect we discussed, and actively trigger mast cells in the gut lining to release inflammatory chemicals like histamine.
So, if you are chronically stressed about your job or your finances, your brain is continuously pumping out CRH. That CRH travels to your gut, weakens the barrier, triggers localized inflammation, and makes the nerve endings incredibly sensitive.
It's a vicious cycle where the stress causes the physical gut pain, and the gut pain causes more psychological stress.
And that increased sensitivity of the nerve endings is known clinically as visceral hypersensitivity.
It is a hallmark symptom of IBS. It explains why a completely normal amount of intestinal gas, or the normal stretching of the bowel as food passes through, which a healthy person wouldn't even consciously feel, registers as excruciating, debilitating pain in an IBS patient.
The volume knob on the sensory nerves has been turned all the way up.
Perfectly said. The threshold for pain is drastically lowered.
There is a really elegant study mentioned that illustrates exactly how these pain signals are modulated. They utilized a technology called DREAD-Ds. Now, DREAD-Ds stands for designer receptors exclusively activated by designer drugs.
Sounds like science fiction.
It sounds like something out of a spy movie, but it is an incredible genetic tool. Scientists can genetically engineer specific cells in a mouse to grow synthetic receptors. These receptors do absolutely nothing on their own. But when the scientists administer a specific synthetic drug that only fits that exact receptor, it acts like a remote control light switch, instantly turning those specific cells on or off.
It allows for an unprecedented level of precision in mapping neural circuits. In this particular study, they used the DREAD-Ds technology to target that specific subset of serotonergic enterochromaffin cells in the gut epithelium.
The cells that use serotonin to rapidly communicate with the vagus nerve?
Yes, when they administered the designer drug to switch those cells off, silencing them, the mice no longer exhibited any pain response to chemical irritants or physical stretching in the colon. The pain signal simply vanished.
They chemically severed the pain pathway.
Completely. But conversely, when they used the technology to artificially switch those cells on, forcefully activating them, the researchers observed a massive increase in visceral pain signals traveling up the spinal nerves to the dorsal root ganglia.
So they turned the pain up.
Yes, and here's the most crucial finding. Manipulating these specific gut serotonin signals in either direction, hyperactivating them or completely silencing them, promoted significant anxiety-like behaviors in the mice.
Wait, so just by playing with the serotonin switches in the lining of the stomach, They literally cause the mice to experience anxiety in their brains.
Exactly. It proves that the gut's sensory signaling directly modulates the brain's emotional state. And this is where the microbiome comes back into the picture as a major contributing factor.
How so?
Clinical data shows that patients with IBS consistently have altered less diverse microbiomes compared to healthy controls. Specifically, they often harbor a higher concentration of bacteria that are highly efficient at fermenting dietary carbohydrates.
Which explains why diet plays such a massive role in managing IBS. If you have more bacteria rapidly fermenting sugars, they are producing massive amounts of gas, which physically stretches the intestines. And if those intestines already have visceral hypersensitivity, that stretching is agonizing.
That is the mechanical basis for why specific dietary interventions are highly effective. The low FODMAP diet, for example, strictly limits fermentable short-chain carbohydrates, certain sugars found in wheat, dairy, and various fruits and vegetables.
By restricting these specific carbohydrates, you are essentially starving those hyperactive fermenting bacteria.
Furthermore, adhering to a low FODMAP diet, or a combined Mediterranean and low FODMAP diet, has been shown to literally alter the patient's microbial profile over time. It reduces the specific bacterial byproducts that are known to chemically trigger the enteroendocrine cells and aggravate that visceral hypersensitivity.
You are removing both the physical gas pressure and the chemical irritants by changing the microbiome. You are taking control of the communication by managing the middlemen.
That is the goal of functional dietary therapy.
Now, I think it is important to draw a clear distinction here. We've talked extensively about DGBIs like IBS, where the communication is flawed, but the tissue itself is largely intact. But that is fundamentally different from inflammatory bowel disease, or IBD, which encompasses conditions like Crohn's disease and ulcerative colitis.
Yes, the distinction is vital. IBD is fundamentally an autoimmune and severely inflammatory disease. In IBD, the immune system is actively attacking the lining of the gastrointestinal tract, causing deep ulcers, bleeding, and severe tissue destruction.
So it's an actual physical wound.
Yes. The root cause of IBD is a complex interplay of genetics and immune dysregulation. It is not simply a gut-brain signaling error. However, the gut-brain axis is critically heavily involved in how the disease behaves, progresses, and particularly how it flares up.
I think anyone with a chronic illness knows that stress makes things worse. But the research maps out the exact biological cascade of how a bad day at work translates into a physical autoimmune attack in the gut.
We see it consistently in clinical practice. Perceived psychological stress in IBD patients is one of the strongest predictors of disease relapse and severe flares. The mechanism relies on that same stress-inflammation loop we discussed earlier.
The CRH from the brain.
Exactly. When an IBD patient experiences acute or chronic psychological stress, the brain releases CRH and glucocorticoids. These stress hormones travel down and act upon the enteric glial cells, the support cells within the gut's nervous system.
And what do the glial cells do once they receive that stress signal?
They transition into a reactive state and stimulate local immune cells, called monocytes, residing in the gut tissue. These activated monocytes then begin to pump out high levels of TNF-alpha,
TNF-alpha.
tumor necrosis factor alpha. It is a fiercely potent inflammatory cytokine. It drives the severe tissue damage seen in IBD. In fact, identifying TNF-alpha's role was such a breakthrough that blocking it is currently the primary mechanism of action for some of our most powerful and effective biologic medications used to treat Crohn's and colitis, such as infliximab.
So the stress actively manufactures the exact chemical that destroys the gut lining. But the damage doesn't stop there. Chronic stress also actively prevents the gut from healing the damage it just caused.
That is perhaps the most insidious aspect. The lining of the healthy human gut is one of the most rapidly regenerating tissues in the entire body. The epithelial cells completely replace themselves every 3 to 5 days.
That fast.
Yes. They do this via stem cells, located deep within structures called the intestinal crypts. However, the data shows that chronic psychological stress pathways traveling down from the central nervous system actually reduce the stemness of these cells.
Stemness meaning their fundamental ability to divide, differentiate, and create healthy new tissue. So a constant state of anxiety sends an electrical and chemical signal from the brain down the vagus nerve that tells the cellular factories in the gut, stop repairing the walls.
You have the stress causing a massive inflammatory fire, and simultaneously the stress is turning off the water to the sprinklers.
It perfectly highlights how deeply intertwined our psychological state is with our physical regenerative capacities. The brain can literally halt tissue repair. And when you realize the sheer power the gut-brain axis has over local disease, it forces you to ask, what influence does the gut have over diseases that originate in the brain?
Which brings us to a major paradigm shift in modern medicine, the gut's role in profound neurological and psychiatric diseases.
I have to say, this next section contains arguably the most shocking, counterintuitive concept of this entire deep dive. When you think of Parkinson's disease, you picture a devastating neurological condition. The classic symptoms, the tremors, the rigid muscles, the loss of motor control, they are all entirely due to the death of specific neurons deep inside the brain.
It is the definition of a brain disease.
But there is a massive, growing body of concrete evidence that Parkinson's disease might actually start in the stomach.
It is a radical departure from traditional neurology, but the evidence is compelling. This concept is heavily anchored in what is known as the brake hypothesis, proposed by neuroanatomist Heiko Brake in the early 2000s. To understand it, we have to look at the cellular hallmark of Parkinson's, the accumulation of a misfolded protein called alpha-synuclein.
Which is usually harmless, right?
In a healthy state, alpha-synuclein is a normal protein present in both the central nervous system and the peripheral nervous system, including the dense neural networks of the gut.
It's supposed to be there, helping neurons communicate.
Correct. But the brake hypothesis proposes that an initial trigger, perhaps an unknown viral enteric infection, a specific environmental toxin ingested in food, or a state of chronic, severe gut inflammation, causes the normal alpha-synuclein proteins located in the nerve terminals of the gut lining to misfold into an abnormal toxic shape.
Wait, let me make sure I'm getting this. A literal stomach bug or something you ate could be the initial spark that causes this localized protein in your gut to mutate.
That is the prevailing theory. And here is where it becomes deeply concerning. Once the alpha-synuclein protein misfolds, it acquires prion-like properties. A prion is an infectious protein that, when it comes into physical contact with a healthy version of the same protein, forces that healthy protein to misfold as well. It creates an unstoppable chain reaction.
So once the fire starts in the gut, how does it reach the brain?
It uses the fiber optic cable. The misfolded alpha-synuclein begins to physically travel retrogradely, meaning backward up the nerve, along the massive trunk of the vagus nerve.
Just creeping up the wire.
It propagates cell by cell, crossing synapses, slowly migrating from the peripheral nervous system in the abdomen directly up into the brainstem. Over the course of years, it eventually reaches the midbrain, specifically targeting the substantia nigra, where it accumulates and causes the mass die-off of the dopaminergic neurons, ultimately resulting in the classic motor tremors of Parkinson's disease.
It's like a slow-moving, silent infection traveling up the communication cable, completely undetected. And the physical, real-world epidemiological evidence supporting this timeline is incredible. There was a massive historical study conducted in Scandinavia reviewing patients who had undergone a truncal vagotomy.
Yes, that study is a cornerstone of this theory.
Because decades ago, before the invention of modern acid-reducing drugs, if someone had severe, life-threatening stomach ulcers, surgeons would perform a truncal vagotomy, which literally involves cutting the main trunk of the vagus nerve to stop the brain from telling the stomach to produce acid.
It was a drastic but common surgical intervention.
Researchers looked back at decades of medical records and found that people who had a complete truncal vagotomy had a significantly reduced risk of ever developing Parkinson's disease later in life. By severing the nerve for an entirely unrelated reason, the surgeons had accidentally cut the bridge to the castle.
The misfolded proteins had no physical highway to reach the brain. It provides striking circumstantial evidence in humans, and this has been replicated and observed directly in animal models.
They tested it directly.
In highly controlled laboratory settings, researchers injected preformed, misfolded alpha-synuclein fibrils directly into the muscular wall of the duodenum, the upper part of the small intestine, in healthy mice. Over the following months, they physically tracked the abnormal protein as it propagated up the mouse's vagus nerve and infiltrated the brain, eventually causing motor deficits.
Wow.
But crucially, if they surgically severed the vagus nerve in the mouse prior to the injection, the spread was halted entirely. The brain remained perfectly healthy.
The disease was literally trapped in the stomach. And this gut origin theory aligns so perfectly with what patients actually experience in the real world. Long before a patient ever develops the classic hand tremor or the shuffling gait that we associate with Parkinson's, they almost universally suffer from severe, chronic gastrointestinal issues.
Yes, the prodromal symptoms, the early warning signs, are heavily gastrointestinal. Severe constipation, dysphagia, which is difficulty swallowing, and delayed gastric emptying frequently appear 10, sometimes even 20 years before the first neurological motor symptom manifests.
Decades before.
The clinical timeline perfectly tracks with the break hypothesis. The misfolded proteins are accumulating, causing localized neural damage and severe inflammation within the enteric nervous system of the gut for decades before the progressive spread finally reaches the critical motor centers of the brain.
That fundamentally changes the future of neurology. If we know the disease spends 20 years brewing in the gut, it offers a massive window of opportunity to detect it through a simple gut biopsy and potentially intercept it before it ever damages a single brain cell.
It changes everything about early detection.
Now, Parkinson's isn't the only major neurological disease where the gut is deeply implicated. What does the research show regarding multiple sclerosis?
What's fascinating here is that multiple sclerosis, or MS, is a severe demyelinating disease where the body's own immune system aggressively attacks the protective myelin sheath wrapped around nerve fibers in the brain and spinal cord. For a long time, the focus was entirely on what was happening inside the central nervous system.
But again, we have to ask, where do those autoreactive immune cells originally come from, and where are they taught to be aggressive? They don't just spontaneously generate in the brain, right?
No, they do not.
Right.
The specific immune cells responsible for the damage in MS are called encephalotogenic T cells. Using incredibly advanced intravital imaging, which allows researchers to watch individual cells move through living tissue in real time, scientists observed the life cycle of these T cells in animal models of MS.
And what did they find?
They discovered that these autoreactive T cells are actually activated and licensed in the lamina propria, which is a thin layer of connective tissue situated just beneath the epithelium of the small intestine.
So the immune cells are being radicalized and armed down in the gut lining.
Exactly. They adopt a highly pro-inflammatory aggressive phenotype while residing in the gut, strongly influenced by the localized microbiome and the specific intestinal immune environment. It is only after they undergo this localized activation in the gut that they enter the bloodstream, circulate, breach the blood-brain barrier, and begin stripping the myelin away from the nerves in the brain.
It totally flips the script. If the immune troops are being trained and deployed from the intestine, then treating MS might eventually involve changing the environment of the gut so those cells never become aggressive in the first place.
That is the hope for future therapeutics.
And this systemic gut-to-brain influence extends far past physical neurology. It reaches deeply into the realm of psychiatry, specifically regarding major depressive disorder and schizophrenia. We've spent decades treating depression as a strictly chemical imbalance originating inside the skull. But there is a study discussed in the research that honestly redefines how we should view mental health.
The study involving fecal microbiota transplantation, or FMT, is a watershed moment in psychiatric research. Researchers wanted to isolate the specific effect of the gut microbiome on mood, so they utilized germ-free mice.
These are mice that are born and raised in completely sterile, isolated bubbles, right?
Yes, they possess absolutely zero bacteria in their gastrointestinal tracts. Their microbiomes are a blank slate.
So whatever bacteria you introduce to them, you know exactly what is causing the reaction.
Exactly. The researchers took a fecal sample containing the complex established microbiome from a human patient suffering from clinically diagnosed severe clinical depression. They transplanted that depressed human's microbiome directly into the gastrointestinal tract of these germ-free mice.
And what happened to the mice?
The results were stark and definitive. Simply receiving the gut microbiota from a depressed human was sufficient to induce a profound depression-like phenotype in the mice. They exhibited anhedonia, meaning a lack of interest in rewarding activities, and severe behavioral despair.
Just from the bacteria.
But it went deeper than just outward behavior. When the researchers examined the brains of these mice, they found significant, measurable inflammatory gene expression changes specifically located in the hippocampus, a key structural region of the brain deeply involved in regulating emotion, learning, and memory.
That completely upends our traditional model. You literally transferred a psychiatric condition from a human being into a mouse via a stool sample. It suggests that depression isn't just a localized problem with serotonin reuptake in the brain. It is a whole-body, systemic inflammatory issue that is heavily driven, or at least deeply modulated, by the bacteria in our gut.
It forces the psychiatric field to synthesize a much broader understanding of these disorders, Altered dysbiotic microbiomes and increased intestinal permeability, that leaky gut allowing microbial metabolites and chronic inflammatory cytokines to continuously reach and bathe the brain, are deeply tied to the pathophysiology of depression, anxiety, and even the severe cognitive symptoms seen in schizophrenia.
And knowing that the gut physically dictates the brain's mood gives us a completely new avenue for treatment. If the wires are sending bad signals, we can try to override the signals. This is why vagal nerve stimulation, or VNS, has become such a powerful tool.
Yes, the clinical application of VNS perfectly demonstrates the power of this anatomical connection. VNS involves surgically implanting a small device similar to a cardiac pacemaker under the skin of the chest. A wire is threaded up and wrapped directly around the left vagus nerve in the neck. The device delivers mild, precisely timed electrical pulses directly into the nerve.
And because the vagus nerve is a one-way superhighway carrying information up from the gut, those electrical pulses travel straight into the brainstem.
Precisely. They travel up the vagal efferent fibers and heavily stimulate areas of the brain that produce serotonin and norepinephrine. VNS is now an FDA-approved, highly effective treatment for severe treatment-resistant depression. Cases where multiple traditional SSRI medications and therapies have completely failed.
By tapping into the gut's dedicated communication highway and artificially stimulating it, we can actively modulate the brain's neurotransmitter levels and drastically improve a patient's mood. We are using the gut's hardware to fix the brain's software.
It is incredible.
We can literally hack the vagus nerve to treat depression. So what does this all mean for us, the listeners, in our daily lives? Especially when it comes to the choices we make 3 times a day, what we decide to eat. Let's examine how the modern Western diet is essentially hijacking this incredibly delicate, highly evolved gut-brain reward system.
To truly understand the current global obesity epidemic, we have to look back at the history of how the medical community originally thought appetite and fat storage worked. It was a remarkably simplistic view. In 1950, a spontaneous genetic mutation occurred in a laboratory mouse colony, creating what became famously known as the obab mouse.
What happened to them?
These mice were uncontrollably hyperphagic. They never stopped eating, and they grew to roughly 4 times the weight of a normal, healthy mouse.
They were just massively obese mice. Why?
Decades later, with the advent of better genetic sequencing, scientists finally discovered the cause. The missing gene in these mice was responsible for producing a hormone, which they named leptin. Leptin is primarily produced by your fat cells, and its evolutionary job is to travel to the brain and signal satiety. It tells your brain, we have plenty of stored energy, you can stop feeling hungry now.
And shortly after, researchers discovered ghrelin, the hormone produced by the stomach that actively stimulates hunger. So you have the I'm full hormone and the I'm starving hormone. The logical assumption then was that obese humans must have a broken system. They must produce way too much ghrelin and not enough leptin.
That was the widely held belief, but the biological reality is the exact frustrating opposite. The clinical data shows that obese individuals actually have significantly higher circulating levels of leptin than lean individuals, and surprisingly lower baseline levels of the hunger hormone, ghrelin.
Wait, really?
It is not a hormone deficiency. The problem is that their brains have developed severe resistance to the leptin. The fat cells are screaming at the brain, "We are full, stop eating!" But the brain has become deaf to the signal. Furthermore, obese individuals frequently have lower levels of other crucial satiety hormones, like GLP-1, being produced by those enteroendocrine cells in the gut.
So the hormonal signaling is completely out of sync. But hormones are only the slow, systemic part of the conversation. The rapid, direct electrical connection between the gut and the brain plays a massive, perhaps even more powerful role in driving our desire to eat. It's not just about volume and energy, it's about reward.
Exactly. The vagus nerve isn't just sending sterile data about caloric density or stomach distension. It is actively transmitting data about pleasure. Researchers mapped these precise neural circuits and found that specialized cells in the right branch of the vagus nerve project their signals straight into the brainstem, which then form direct synaptic connections with dopaminergic neurons in the substantia nigra and the ventral tegmental area.
These are the deep, primal reward centers of the human brain. So when food hits your stomach, the vagus nerve triggers a release of dopamine, the feel-good chemical, making you want to do it again. But different foods trigger different pathways, right?
Yes, and this is where the evolutionary biology clashes violently with our modern food environment. When you consume dietary fat, it activates one specific, dedicated signaling pathway in the vagus nerve. When you consume sugar, it activates a completely separate parallel pathway, specifically utilizing those lightning-fast neuropod cells we discussed earlier, which fire utilizing the neurotransmitter glutamate.
I want to paint a vivid picture for you, the listener, of what this means when you walk into a grocery store. In nature, foods that are naturally high in both fat and sugar simultaneously are incredibly rare. You have fatty foods like nuts or avocados, and you have sugary foods like berries or honey, but they almost never occur together in high concentrations. But in the modern Western diet, we have engineered food to do exactly that. It's a glazed donut. It's a massive chocolate milkshake. It's a slice of highly processed pizza.
And the physiological consequence of combining them is staggering. When you eat a food heavily engineered with both fat and sugar, it doesn't just activate one pathway. It fires both the fat-sensing pathway and the sugar-sensing neuropod pathway simultaneously.
Both at once.
This dual activation creates a massive synergistic effect, resulting in a completely unnatural, overwhelming surge of dopamine release in the brain's reward centers.
It is the biological equivalent of a slot machine paying out the jackpot. The gut is literally overriding your conscious, logical willpower. You might cognitively know you shouldn't eat the second donut, but your gut is hijacking your fundamental evolutionary survival circuitry, screaming at your brain, "We found the ultimate rare energy source. Keep eating, do not stop. Survival depends on it."
It is a profound evolutionary mismatch. Our biology evolved over millions of years to survive in an environment of extreme caloric scarcity. It is designed to aggressively seek out and reward the consumption of dense energy. But we now live in an environment of hyperpalatable, heavily engineered, inescapable abundance. The biological hardware simply hasn't caught up to the supermarket.
And those neuropod cells are incredibly sophisticated. You can't just trick them by drinking a diet soda instead of a regular soda. The cells in your gut actually know the difference between real calories and fake ones.
They do, and the mechanism is fascinating. The neuropod cells possess distinct receptors. When they detect genuine sucrose, real sugar, they activate the vagus nerve using the excitatory neurotransmitter glutamate. But when they detect artificial non-caloric sweeteners like sucralose, they respond by utilizing a completely different signaling molecule, ATP.
They use a different chemical language entirely.
Through repeated exposure, the brain's reward centers learn to distinguish between the glutamate signal, which equals real energy, and the ATP signal, which equals a caloric trick.
The gut literally knows you're trying to fool it with a packet of sweetener.
Yes, this is why, on a subconscious physiological level, your body will consistently develop a strong preference for the real sugar over the artificial sweetener. The gut-brain axis is specifically actively wiring your behavioral preference toward the actual usable calories, regardless of your conscious dietary goals.
It is brilliantly, frustratingly smart. And understanding this exact hyper-specific circuitry, knowing exactly which receptors and transmitters are involved, is what has finally led to the development of the modern miracle drugs we are seeing everywhere today. We are finally targeting the gut-brain communication directly. Let's look at how these drugs actually work, starting with a medication called linaclotide, which is used to treat the severe pain of IBS.
Linaclotide is a prime example of precision pharmacology targeting the gut-brain axis. It's classified as a guanylyl cyclase C agonist. Remember earlier when we discussed visceral hypersensitivity in IBS? The excruciating pain signals were originating from those hyperactive, overly sensitive sensory neurons and neuropod cells in the gut lining.
The ones where the volume is turned all the way up.
Yes. Linaclotide works by binding specifically to the GUCY2C receptors, located directly on the surface of those excitable cells in the intestine.
So it docks right onto the cells that are causing the problem. What does it do once it binds?
It effectively acts as a localized dampener. It initiates an intracellular cascade that quiets the electrical excitability of the neuropod cells. By calming the local gut cells, it dramatically reduces the number of pain signals firing up the spinal afferents to the dorsal root ganglion.
You are stopping the pain signal at its absolute origin point before it ever has a chance to reach the brain.
Exactly. Clinical trials show that over 50% of IBS patients treated with linaclotide report a significant life-changing reduction in abdominal pain.
That is incredible. And then, of course, we have the absolute blockbuster medications dominating the cultural conversation right now. Semaglutide, tirzepatide, and the broader class of GLP-1 receptor agonists used for treating obesity and type 2 diabetes. It feels like everyone knows someone who has completely transformed their health on these medications. They're routinely producing weight loss results of over 15% to 20% of total body weight in clinical trials, which was previously unheard of outside of bariatric surgery. But how do they actually achieve this through the gut-brain axis?
To understand their efficacy, we have to look at what GLP-1 actually is. Glucagon-like peptide 1 is an endogenous incretin hormone. It's naturally produced and secreted by those enteroendocrine cells in your lower intestine in response to food consumption. GLP-1 agonists like semaglutide are synthetically engineered versions of this hormone designed to last much longer in the bloodstream without being broken down by enzymes.
So you are basically flooding the system with a synthetic version of the I just ate hormone.
Essentially, yes. These drugs bind tightly to GLP-1 receptors, which are widely distributed. They do have localized effects in the gut. They significantly slow down gastric emptying, meaning food literally stays physically sitting in your stomach for a much longer period, creating a prolonged physical sensation of fullness.
But the real power is in the brain.
But their most profound effects are neurological. They bind directly to the receptors on the endings of the vagus nerve in the gut, and they also cross into the brain via those circumventricular organs to bind directly to receptors in the hypothalamus and the brainstem.
They are essentially taking over the entire communication network. It's like a chemical megaphone situated in the gut, constantly shouting, "We are completely stuffed, stop eating," directly into the brain's hunger centers 24 hours a day.
It is a massive, relentless satiation signal. What is particularly fascinating from a neurobiological perspective is that the research indicates there are parallel, independent systems at work here. The vagal neurons projecting from the gut that possess GLP-1 receptors do not seem to overlap with the specific neuronal pathways that prompt the brainstem itself to release its own centrally produced GLP-1.
What does that mean in terms of how the drug works?
It suggests that there are parallel peripheral gut-to-brain pathways and independent central brain-only pathways that both function to suppress appetite. By utilizing a drug that circulates systemically, you are simultaneously co-opting both of these multiple redundant physiological pathways at the same time.
Hitting it from every angle.
This dual action achieves an additive, overwhelmingly massive suppression of both physiological hunger and the psychological food noise or constant craving for food.
And because these GLP-1 receptors are scattered throughout various regions of the brain, not just the appetite centers, researchers are seeing these drugs do things entirely unrelated to weight loss.
Exactly. Because they are broadly modifying reward circuitry, dopamine response, and significantly reducing the central neuroinflammation, these GLP-1 medications are currently showing massive, incredible potential in clinical trials for treating a wide variety of brain-based disorders.
Like what?
We are seeing data suggesting they can slow the cognitive decline in Alzheimer's disease, reduce the cravings associated with severe alcohol use disorder, and even improve outcomes in cardiovascular disease independently of the weight loss. We are using a synthetic gut hormone to structurally and chemically protect the brain.
It is an absolute fundamental revolution in how we practice medicine. We have gone from thinking the stomach is just an unglamorous, acidic bag that mechanically mashes up our lunch to realizing it is a massive, highly sophisticated sensory organ. It is a complex hormonal factory, it is a primary training ground for our immune system, and it is a direct, hardwired puppeteer of our mood, our anxiety, and our most fundamental daily choices. Which brings us to our wrap-up. We have taken an incredible, deep biological journey today, tracing the signals from the very lining of the intestines, up the wandering vagus nerve, and straight into the center of the brain.
It is a profound anatomical connection. And this raises an important question, almost a philosophical consideration. When you look closely at the sheer volume of data, hormones, and electrical impulses flowing upward from the gut.
It makes you wonder.
If our resident gut bacteria are actively dictating the production of the hormones that make us feel hungry, anxious, or depressed, if the specialized neuropod cells in our intestines are subconsciously, chemically steering our behavioral choices towards sugar just to satisfy a localized dopamine circuit, and if our brains are literally encoding physical, actionable memories of peripheral gut inflammation, it forces you to question the nature of autonomy.
Right.
Where does the conscious you actually begin and end? When you experience an intense craving for a specific food, or when you feel a sudden, unexplainable wave of profound anxiety, is it truly your conscious autonomous brain making a top-down choice? Or are you, to a surprisingly large extent, simply a conscious passenger responding to the relentless electrical and chemical demands of millions of specialized gut cells and trillions of foreign microbes, all actively lobbying your brain for their own survival?
That is a deeply humbling thought to chew on the next time you get a strong gut feeling. This paper is published under a Creative Commons Attribution 4.0 license, and full credit is provided in the episode description. Thank you for listening to One Paper Orbit Day, our research paper podcast. Our goal is to make peer-reviewed research accessible to everyone. If this episode was helpful, please share your thoughts and let us know which papers you'd like us to explore next. Thank you.